Hey everyone, it's Miss Bell. So this unit is on genetics, and so genetics is going to start out with Mendel, who is the father of genetics, and heredity. Now you might remember what heredity means, but it's the passing of traits from parents to offspring. So if you look here, that's why we have pictures of little babies, because these babies would have received their traits from their parents. So that brings us to just some basic terminology. Genes are little pieces of chromosomes that are responsible for all of your traits. So we've already talked about genes some, so this should really be more of a review for you. But if you recall, you have 46 total chromosomes in 23 pair. But on each one of those 46 chromosomes, you have thousands of genes. So one gene is usually going to control one trait. Your traits are going to be things like things you see on the outside, like eye color, hair color, skin color, and height. But it's also going to be things on the inside too, like whether or not you have the normal or abnormal gene for certain diseases. Like we've talked about the breast cancer gene a lot. You either have the normal gene there or you have the breast cancer gene. Hopefully, you don't have the breast cancer gene. But there's lots of other genetic disorders, and you either have the normal gene or the gene for the disorder. So anyway, we've already been talking about genes and chromosomes. So now this unit is about how you inherit the traits, the genes that you get. And so that is why we call it genetics. So it's how genetics is how you inherit the things that the genes that you get. So again, we have a picture here of a very pregnant belly, and you can see the little foot there. My belly never did that. I guess I'm too fat. I don't know, but yeah. So this is Gregor Mendel. So when you're talking about genetics and heredity, this is the first name that's going to come up. So Gregor Mendel is considered the father of genetics. So genetics is the study of heredity, and heredity, like I said earlier, is the passing of traits or characteristics from the parent to the offspring. So, for example, my daughter, Gemma, she has dark hair, which she would have got from both myself and Mr. Bell. And she has dark eyes. She has Mr. Bell's eyes rather than my eyes. So he passed that trait on from him. So you can attribute all of the person's traits to one of the parents. It might not directly be from the actual parent, it might be from a grandparent because you, as a parent, have the traits that your parents had. So, anyway, Gregor Mendel, he was the father of genetics. So, he was in Austria, which is in Europe, and he was a monk. So, this is back in the 1800s. He doesn't look like a monk that you're thinking of, probably. Most people think of Tibetan monks that are bald and they wear the robes. But he lived in a monastery. So you can think of a monk as like the male version of a nun. They are men who devote their life to their religion. But he didn't just solely study his religion, he also studied genetics at the monastery. So, anyway, he came up with a lot of based on his experiments that he did, he came up with a lot of some basic genetic principles that we're fixing to talk about. Specifically, he came up with the whole dominant recessive thing. Anyway, so again, a trait is a characteristic you inherit, like blue eyes, brown hair, blonde hair, curly hair, straight hair. Those things are traits. So that brings us to monohybrid crosses. So first off, mono, we know, means one. And hybrid is going to be referring to like different differences. So a monohybrid cross is a cross involving one pair of contrasting traits. Okay, so like the most common thing we see would be tall or short. Those are contrasting. They're opposites. But they're still the same trait. The trait is for height. Okay, so that's what I mean when I say one pair of contrasting traits. So a monohybrid cross is going to cross one particular type of trait, like height. It's either tall or short. So anyway, a monohybrid cross is a 4-square Punnett square like you see here on this slide. So what happens is for every inherited trait that you have, you get 2 copies of the gene. One from your mom, one from your dad. And we've talked about this before. These genes are called alleles or alleles. So anyway, your alleles are simply the letters we use in the Punnett square. Okay, so this is an allele and this is an allele. Okay, so these are the gametes from the male parent. Remember, gametes are sex cells. Okay, so this would have been in one sperm, this would have been in another sperm. Okay, so if you recall, your sperm and egg, your sexually reproducing organisms, are going to be haploid. Okay, remember, gametes are haploid. So they're going to have half of the normal number of genes or chromosomes that you normally would have. So on the outside of the Punnett square, it will always be the gametes from each parent. And it doesn't matter if you put the male on the top or the male on the side. The female on the top, female on the side. It doesn't matter. As long as this side you have one parent and this side you have the other parent. But anyway, here you have the gametes from the female. So these are the 2 possibilities that could go in an egg. On the inside of the Punnett square, these are their babies. So these are the 4 possible babies you could get from crossing this particular female with these genes with this particular male with those genes. So this is a monohybrid cross. So again, the monohybrid cross is really just your basic 4-square Punnett square. And so you're looking at one particular trait. So in this Punnett square, they crossed a tall pea plant with a short pea plant. Peas like the peas that you eat. So in this particular case, big T is tall and little t is short. So they crossed a short with a tall, and all of the offspring, 100% of that offspring, are going to be big T, little t. And because the big T is dominant, that's going to be tall. And so we'll get into that in a minute. So, some more terminology here. So, dominant, I think a lot of people have an understanding of what dominant means. But dominant is the expressed form of the trait. Okay? So that means you will see it. If the dominant is present, you will see that dominant trait. So what that means is if you have a big letter, a capital letter, then you will see that particular trait. So if the capital letter stands for tall and that baby has a big T, it will be tall. It is dominant. That is what you will see. The dominant letter is always written first. So if you have a big T and a little t, the big T will always be first, just like in a sentence or if you're writing a word. Recessive. Recessive is not expressed if the dominant is there. So it is called the hidden trait. Okay, so the only way you see the recessive is in when there is no dominant present. So the only way I would see a recessive would be little t, because there's no big T's, there's no talls. And little t would code for short. So the only way you would see short is if you had both genes for short, or you only had genes for shortness. So anyway, what this is saying on the bottom is that for all these contrasting forms of a trait, you'll usually have a dominant and a recessive form. So, some other big words you need to know. Phenotype and genotype. Okay, so for phenotype, you need to think phenophysical. Okay, so this is what the way it looks. Okay, this is what you can see, usually on the outside, like if it's hair color or eye color or something. If you have the disease or you don't have the disease. So anyway, if you're looking at, if I say big T little t equals tall, then tall is the phenotype. Now, the genotype is your genes, okay, or your alleles, or we can also say it's the letters, okay. So, big T little t would be my genotype, whereas tall would be my phenotype. So anyway, genotype is going to be your genetic makeup. If you know the genotype, you definitely know what the phenotype is. I know that big T, big little t are going to be tall. Little t are going to be short. Now, I do want to say that that's not always the case, but it is a very common example that we see. So you really have to read the example because sometimes short is actually dominant. But anyway, if I see the letters here, then I know for sure what it's going to be. If I'm only given tall, I don't know what it's going to be. It could be big T, or big T, little t. So I can't tell the genotype by simply looking at if it's tall or short. Now, that being said, if it's short, there's only one genotype that's short. So yes, I could see that. But I wouldn't know the dominant genotype. So, anyway, the genotype codes for the phenotype. So, other important terms to know. Homozygous and heterozygous. So, one of the prefixes that we learned before is homo. Homo means same or similar. Okay, so as far as genetics goes, homozygous, zygo would be like the zygote. So it's referring to the alleles. So homozygous is when you have two alleles for the trait that are exactly the same. So what that means is it's either big T, or little t. Okay, because the big T, so this would code for like tall, and then that would code for tall. So tall, they're the same. This one would code, that little t would code for short, and this little t would code for short. Short, they're the same. Okay, so this is also known as a purebred or true breeding plant. So homozygous, another word for that would be purebred or true bred. So if you see homozygous, it will have the same letter. Now, it might be homozygous dominant or homozygous recessive. So you need to think homo, same letter, dominant, big letter. So homozygous dominant, that would be big T. So short plant, same thing. Homozygous recessive, homo would be same letter, recessive would be little letter, so two little letters. So, when you're looking at Punnett squares and doing Punnett square problems, sometimes it might say cross to homozygous recessive with a homozygous dominant. Or it might tell you to cross a big T with little t, and then ask you what percent of offspring are homozygous. So you have to be familiar with the terms and be able to go back and forth between the alleles, the letters, like big T, and little t, and their descriptors like homozygous dominant, homozygous recessive. And sometimes they might say like homozygous for short or homozygous for tall. So you really have to look at the context clues and read to find out what is the dominant and what is the recessive. So be very careful of that. So here we have brown hair versus blonde hair. Does this remind you of anyone in our class, these pictures? I can think of a few people that might resemble these guys. Okay, so then you have hetero. So heterozygous means different alleles. Okay, so different letters. Okay, so for a typical genetics, so in normal genetics, heterozygous is always going to be one big letter and one little letter. And it's hetero because it codes for, well, it's two different letters, but it's coding for two different traits. Okay, the big letter is going to code for tall in this case, and the little letter is going to code for short. So in this case, the individual will be tall because the little letter, the short, is hidden because that's the recessive. So with heterozygous, another name for that is a hybrid. Okay, so you might be told, given a question saying to cross two hybrids. So you know you would cross a big T little t with another big T little t. So, again, the only option really for this case for hetero, it's two different ones, is a big and a little. One big, one little. So if you take a look at this picture, I really like this picture here. You have two of the same, big A and big A. And here you have two different ones, big A, little a. Okay, so if you notice, these both code for the same color blue. Whereas this one codes for a darker blue and this one codes for a lighter blue. Okay, so as you can see, these code for different traits or contrasting traits, and these code for the same. So this would be homodominant, and this would be heterozygous. So basically, whenever you're doing these Punnett square problems, you're going to have three options. You're going to have two big letters, a big and a little, and then two little letters. So these two have the dominant letter, so they're going to display the dominant trait, whatever that may be. And this is going to be recessive because there's no dominant letters, so their only option is to display the recessive trait. So whenever you are working these Punnett square problems, that took me seconds to do that. So I would highly recommend you creating yourself a little key like this. And so like I'm going to give you another example. So let's use B. So for B, I have three options. Either too big, a big and a little, and little. I know these both are going to have the dominant. Okay, and let's say, let's do brown eyes, blue eyes. Okay, so let's make brown eyes dominant and blue eyes recessive. Okay, so I would just have to read the question to figure that out. But this key, do this for every time you do a Punnett square. It really, truly helps. When we get to the bigger 16 square Punnett squares, it will become very difficult for you if you do not do that. Okay, so we were talking about Mendel. So what did Mendel do? So again, he was an Austrian monk. He was the first guy who really studied heredity and genetics. And his studies were so good, they're still basically used today. And he's credited with coming up with dominant and recessive genes, the idea of dominant and recessive genes. So that's known as the law of dominance. But anyway, so in Mendel's experiments, what he did, he grew a whole bunch of pea plants, basically. At the monastery, he had all these pea plants that he grew and he tended to. And so in his first experiments, he used monohybrid crosses because he was only looking at one particular trait, and that particular trait was the plant's height. And that's one reason why you see a lot about big T is tall, little t is short, because that's based on Mendel's experiments. But anyway, so he crossed tall plants with short plants. So we're going to talk about what he did at each step in his experiment. Okay, so there's three main steps, and you do need to know these three steps. So make sure that you understand each of the three steps. So they're named for their generation. So step one, he created the P or parental generation. So P1, parental generation. Okay, so how he did that was he created purebreds. And he did that through self-pollination. So if you think about a plant, if it has flowers, it's going to be sexually reproducing. So if you have a plant and it has multiple flowers on it, the whole plant, the whole stem, if this is your plant and you have a flower here, and then a flower here, and then a flower here, and just excuse my horrific drawing, and a flower here. You got all these flowers on this plant. Well, the pollen from this flower could go to this one, but it's on the same actual plant, so it's going to have the same DNA. So that's called self-pollination. It's still going to produce haploid gametes, though. So you're still, but it's within the same genes. So pollen is transferred from one flower to another flower, but it's on the same plant. That's self-pollination. So it's pollinating itself. So anyway, in his first step, he ensured that all he had was purebreds. Okay, so that means they were all homozygous. That was his goal, was to make sure that every plant that he had was either homozygous for tall genes or homozygous for short genes. He knew for sure that they had only tall genes in the tall plants and only short genes in the short plants. And what he did was he kept making just a whole bunch of generations of these plants until all the offspring were only tall in this one group. And then in this other group, all the offspring were only short. So this is called the parental generation. He self-pollinated to have all homozygous. So then he has his step 2. So in step 2, what Mendel did was he cross-pollinated the parents from the parental generation. So in the P1 he would have produced a solid purple or a solid white. So they would be homozygous. Then in the F1 he crossed them. Okay? And when he crossed this all homozygous dominant and all heterozygous homozygous recessive, he got all heterozygous individuals. So they all would have a big and a little letter. Okay, so notice how I'm putting the line above the P, because a big P and a little p look very similar. So in putting the line above it, I know that it's lowercase. You can also do what we call a tick apostrophe for a short one, just if you're using letters that the capital and lowercase are very similar. Anyway, so step 2, this is called the F1 or first filial generation. Mendel cross-pollinated the two parents. So he cross-pollinated his homozygous tall with his homozygous short. And he came up with all heterozygous offspring. And that leads us to the last step. So in the last step, he took his heterozygous and he self-pollinated it. Okay, so the Punnett square would be a big little crossed with a big little. And when he did that, they called it the F2 generation, or second filial. Filial means family. So when they looked at what was left of this cross between the two heterozygous, they noticed that 75% displayed the dominant trait. Either the purple, like you see in this picture, or the tall. But 25% showed the recessive trait. Now, mind you that the parent is not recessive at all. The parent is purple. So you crossed a purple and you got a white. Okay, or you crossed a tall and you got a short. Okay, so this is where recessive came in. This is the key part that allowed him to be able to understand dominant and recessive genes. So, step 3, F2 generation, he self-pollinated his hybrids. And he came up with, when you do that, so if you did it with T's, you would have one homozygous dominant, you'd have 2 talls that are hybrids, so 3 talls, and then 1 short. So you'd have 1 or 25% that are short. So just to review, the P1 or parental generation, that's your homozygous tall and your homozygous short. Okay? And then in the F1 generation, he crossed those two and produced all hybrids, all heterozygous. Sorry. And then in the F2 generation, he crossed those hybrids and he got 75% tall, 25% short. So he did have the recessive trait reappear. So it skipped a generation. So that recessive short, it skipped this second generation. So this is just another picture showing that if you cross-pollinated the parents in the F1 generation, you would have all hybrids. And then in the F2 generation, your recessive would show back up. And so if you notice, you skipped a generation where you had no recessives.